Hi students, this is MNR from the Department of Botany. Today we are going to discuss about a topic microbes in human welfare. What is the topic? Today's topic is microbes in human welfare. First we better to know the complete meaning of the topic. What actually we are going to discuss in this topic. So, you all please pay right attention on this topic what it is discussing. So, see students, microbes. Microbes are a very small, invisible, tiny particles which they are playing major role and major components in the biological systems present on the earth. You know. So, these microorganisms are playing a pivotal role on this earth surface. Generally, people are thinking about these microorganisms as harmful, dangerous, dreadful components. But dear students, some of the microorganisms are only dangerous, dreadful, harmful, which they are going to cause some diseases. At the same time, we know very well, some of the microorganisms are very much useful to us. In our everyday life, we depend on those organisms for all the living activities, what we are going to get some useful things or products and comfortable life to lead our life comfortably. What are the factors or advantages we are obtaining from the microorganisms we are discussing in this topic. So, microbes, you know very well. One more time we better to discuss about these microbes, what actually they are. So, microbes are nothing but microorganisms, you know very well. Besides macroscopic plants and animals, we are also observing so many very small invisible particles on the earth. Sir, they are invisible particles. How do we visit them? You know, to visit, to watch, to observe those invisible particles like microbes, we are using a device called microscope, which is going to magnify its size thousands of times, increases. So, by using that microscope, we can easily to visit those microorganisms which are not visible to naked eye directly. That's why we should depend on the device microscope. So, coming to the point. So, microorganisms generally we observe bacteria, some fungi, viruses. You know, dear students, in the viruses, we are observing some other components are there like, viroids and prions, which is also come under the category viruses. So, like that, so many small, tiny, invisible particles are there along with the macroscopic plants and animals, which they are present as major components in the biological systems on Earth's surface. So, today we are discussing about all these are the living components or microbes. How do they help us? How do they play pivotal role in the human welfare? What are the advantages we are obtaining from these microorganisms is the main important concept what we are going to discuss in this chapter, microbes in human welfare. So, before going to discuss about all the subtopics which are present in this chapter, we better to know little bit previous knowledge on these particular microorganisms. Students, in the class 11, you already learned well about the diversity of living organisms. or diversity in living organisms. In that, taxonomy, classification, different kingdoms given by different scientists, So, in that, in which kingdoms generally we are observing the microorganisms are present. The microorganisms are inserted, or that microorganisms are kept in which kingdoms in the different classifications, different kingdoms of classification given by so many scientists. You know very well. Dear students, for example, the bacteria belongs to kingdom, Monera, ancient kingdom. Okay, right, fungi, as it is that kingdom called as fungi. Okay, next, viruses are the separate category of organisms because they are biological entities. They are not truly living and not truly non-living. Because viral particles are the particles which are only that nucleoprotein particles which they are non-living when they are outside the living cell, but they are living when they are present inside the living cell. Along with all these living organisms like bacteria, fungi, viruses, we also discuss about, we need to talk about some, right, members of protozoans. Protozoa. The members of protozoa belongs to the kingdom Protista. So, all these are the different components as microorganisms we are discussing. Okay, how do they help us? How do they play pivotal role in the human welfare? Right, Nana? So, now we are discussing about that presence, habitat, where they are present. Right, microorganisms present everywhere. Microorganisms present everywhere. Right, especially you may take first component in the microbiology, that is bacteria. Bacteria generally we called as the bacteria generally called as organisms which present everywhere. So, they are called as ubiquitous organisms. Ubiquitous, omnipresent, or cosmopolitan organisms. Cosmopolitan organisms. So, this is the information about bacteria. In the same way, all these are the living organisms are present everywhere. Wherever you may notice, want to check them, they are present. Okay, so suppose they are present in the soil, they are present in the water, they are present on the surface of the body of plants and animals, inside the body, everywhere. Though we are not expecting living organisms and their living conditions in which habitat we may not expecting to continue their life. In those conditions are also these bacteria, these so many microorganisms leading their life normally. Okay, means under extreme conditions these microorganisms can leading their life normally. So, for example, right, some bacteria they are living under the extreme conditions like extreme hot springs, very hot temperature. The temperature maximum is as above 100 degrees Celsius. In that conditions also these bacteria can lead their life normally. Okay, so those are called thermophiles. Some bacteria, they are living, they sustain their life, they can lead their life normally in the more acidic conditions. Those are called halophiles. Some bacteria, they are living on the surface of the body of organisms, plants and animals. Some are, right, that's why these bacteria possess a simple structure but complex in behavior. Generally, the bacteria has simple body structure but a complex behavior. So that is the simple information about all these microorganisms. Whatever the organism is present on the earth, they are playing pivotal role. How do they play pivotal role to human life and human welfare? What are the advantages we are obtaining from these microorganisms for our better life? Okay, students, now coming to the subtopics which we are going to discuss in this lesson. Okay. So, in this chapter, microbes in human welfare, we are going to discuss about subtopics is as microbes in household products, and next one is microbes in industrial products, microbes in sewage treatment, microbes in the production of biogas, microbes as biocontrol agents, and some microbes we used as biofertilizers. So, of course, microorganisms are not only giving these useful or advantages what generally we are obtaining from microorganisms, but they are also giving so many useful or advantages things they provided to us. But in this chapter, we are going to discuss about all these 6 different levels of aspects we are discussing, which are very much helpful to lead human life better. Okay, dear students. So, now we better to discuss brief discussion about all these 6 various types of advantages or the subtopics what they given in this chapter. Microbes in human welfare. In that first one, microbes in household products. Microbes in household products. Regularly from the early morning to night, just we are doing so many life activities. In that, dear students, to lead all these life activities, to lead our life normally by all these living activities requires food. So, in the food, we are using so many microorganisms, but we don't know exactly their presence in all the food. In so many foods, how we are using microorganisms. Have a look into the microorganisms, how do they play a role in our foods what we are preparing. So, whatever the food we are obtaining from our home or from our kitchen, but indirectly, invisibly, so many microorganisms they entered and give more flavor, texture, taste, and whatever the food is, we are taking with much very interest. So, in that microbes in household products, regularly in that first important thing, it is curd. In our everyday life, we are using curd. You know very well the importance of curd. So, the curd is from milk. How the curd prepared, how the curd formed from the milk. So, right. Some microorganisms, especially like bacteria, Lactobacillus delbrueckii is a bacteria which play pivotal role in the conversion of milk into curd. In our daily life, we must use curd as a food, one of the foods what generally we are obtaining. So, the preparation of curd, how it done, I think, right? Just we are going to take some amount of, little amount of curd, what we are adding into the fresh lukewarm milk. Then that is the temperature which is present in the milk is suitable for growing more bacteria. Already I said bacteria, Lactobacillus bacteria. Lactobacillus delbrueckii is a bacteria which is going to convert the lukewarm milk into curd. Understood? So, like that microbes in industrial products. So, industrially we are going to discuss about many products. We are obtaining many products. What actually they are once you see. Microbes in industrial products like yes, alcohol. The production of alcohol is done by these microorganisms. They play pivotal role in the production of alcohol in beverage industries. And also we are obtaining from so many chemicals, so many enzymes, so many bioactive molecules, so many medicatory substances like antibiotics. All those are only in industrial scale we are obtaining by using microorganisms. Without microorganisms it is not possible to get all the different types of products what now we are obtaining. Now what we are utilizing. Okay students. So next one is microbes in sewage treatment. Okay, I think right, sewage treatment. What is sewage treatment? Okay, sewage treatment is nothing but every day we are collecting wastewater from the various municipalities. Just we are collecting wastewater, contaminated water from all the household or domestic areas. Just simply they are releasing water into the drainage system. Okay, from all the towns and cities that municipalities water, it is coming into the drainage system. Just we have to collect it and reduce the pollution, reduce the contamination of pathogens which naturally they are present in the, which generally present in the sewage water. Understood? So we have to take the drainage water or municipal water and we have to reduce the contamination, pollution, pathogens, by using some bacteria and fungal members in all the sewage water treatment plants, STPs. Sewage water treatment plants or sewage treatment plants. Understood? So, through that only we are going to reduce the contamination of or reduce the pollution, then only that particular contaminated water releases into the natural habitats. Understood? The next one is microbes in the production of biogas. You know what is biogas? The biogas is also called as gobar gas. Gobar means dung. Okay, here the main or chief raw material for the production of biogas is dung, cow dung, buffalo dung, yes, various different excretory materials what generally as a dung we are using here, and also some grass, dry grass, straw, whatever the waste material, just simply we are biological waste material simply we are adding into this a plant that is a biogas tank. Into the biogas tank only, from the biogas tank only these some microorganisms are going to be added, right, especially like methanogens, methanogenic bacteria. So methanogenic bacteria generally it is present in the dung. How it is present in the dung? Right, in the digestive canal, digestive system of ruminants like cows, buffaloes. Naturally it is present. And it is very much useful for digest the complex materials, understood, cellulose like, understood. So then it can easy to digest and come out of the excretory material like dung. So that is the bacteria naturally present in the cow dung and some other materials which they are easy to anaerobically digest the materials and release energy to increase their number. During the process of anaerobic respiration what done by different methanogenic bacteria, then they are going to release a gas that is called biogas. Biogas is not a single component of gas. It is a mixture of gases. Generally we see more amount of methane and carbon dioxide. Next one is hydrogen. So all these three main or chief gases which they are going to form a particular gas, it is a mixture of gas, it is biogas or gobar gas, students. Next one is we are discussing about that. Next, microbes as biocontrol agents. What is biocontrol agents? Right, dear students? Everywhere farmer, what duties they are doing for giving food to all of us, But these cultivation may not give proper sufficient food to the farmer, though he is making food to us. But what happened, that particular crops, okay, generally they are infected by so many pests, insect pests. So all these are the pests and insects which they are damage the crops. Understood? How we eradicate, how we control them, how we are going to reduce their population to destroy the crops, just we are following to give some chemicals, okay, some chemicals which we are obtaining from some microorganisms. Okay, best example, you know, right? Okay, bt, you know, right? Bacillus thuringiensis is a bacteria what is going to give a particular crystals forms. Okay, the crystals what just we are obtaining and collecting in the form of spores, that spores are going to, okay, damage the living organisms like okay, pests, or what the insect pests are destroying crops, then they can easily naturally control these pests and insect pests. Otherwise what happens, the farmer simply they are using so many, okay, chemical fertilizers. If the farmer continuously using chemical fertilizers, what happened, you know, if he is using continuously these chemical fertilizers, the land after 30 to 40 years it is completely become barren, wasteland, because these chemical fertilizers are going to damage the fertility of the soil by adding injecting different non-biodegradable products and very dangerous and toxic metals, toxic chemicals into the soil which are damage the fertility of the soil. Then that soil is not give the chance for growing plants, not only plants, whatever the microorganisms are useful to the plants, they may not continue their life there because the toxicity increases. So that's why in this scenario we are the people who are going to use to control all the insect pests. Biologically we are using some biological organisms like bacteria, okay, and some other what we call living organisms like okay, microbes, what generally we use to control the insect pests naturally. Then when you are, if you are using microbes to control those insect pests, they may not decrease the fertility of the soil. Okay, they may not okay, change the fertility of the soil, and the soil may not turn into barren land or wasteland. Next one is microbes as biofertilizers. You know, biofertilizers in the place of chemical fertilizers. If you continuously a farmer uses chemical fertilizers, what happened, you know, complete fertility of the soil is going to be decreases. Then it is not useful to sustain the, okay, useful flora and fauna in the soil. So that's in this situation, just we go for the usage of microorganisms which are going to increase the fertility of the soil by adding some organic constants, okay, and inorganic constants like nitrogen fixation. Understood? And maintain porous properties, porous nature in all the soil particles for imbibe the water and percolate the water and for the exchange of gases, everything. Whatever the fertilizers we are obtaining from biological organisms like bacteria. Understood? So simply we are discuss about all the things in the microbes as biofertilizers. Understood? So all these are the 6 topics we are going to discuss, Nana. Okay, so now let us know something about one by one topic which is there here. Understood? Okay, microbes in household products. Here we must focus our concentration on, okay, some important categories of useful things what we are generally obtaining from microorganisms as household products. In that first one is okay, convert milk to curd. It is the first case, okay, in which we are using microbes for getting curd. Next one is just fermented duff. Duff means, you know, right, okay, flour, wheat powder, or some other pulses, what grains what we are using to make idli, dosa, like all those are the different food items what we are preparing. That is we are calling, okay, duff, right? So how we are going to ferment the duff? By using some microorganisms, understood? So that is also very important. So fermented duff we are using for the preparations of idli and dosa, etc. And some microorganisms also we are using for, yes, along with these two we are also using these microorganisms for the preparations of, right, okay. Toddy. What is toddy? In the preparations of toddy, toddy means what? Palm sap. Palm sap. Okay. Palm sap just we are obtaining from the palm trees. Palm trees, right, that is the in South India people, okay, maximum they are utilizing as used as a drink, okay, from all the palm trees like Borassus palabellifer, okay, like different kinds of palm trees, okay, from that only they are collecting some liquid, that is we are calling palm sap, that is generally we are calling what? Toddy. So in the toddy, just we are observing some bacteria play pivotal role to ferment the toddy to give taste, understood? So that is in the preparations of toddy, and also in the preparations of oldest and ancient, okay, food item, that is generally we are observing in all the villages. Nowadays we are getting through large industrial scale, they are also preparing. This is the oldest ancient food, that is cheese. In the preparation of cheese, okay, soft cheese we selecting more, understood? And taste, texture, flavor, in all the three cases, especially texture, flavor and taste, just we are using some microorganisms in the preparation of cheese. Okay, here two different kinds of cheese we are generally observing, that is Swiss cheese and next one is Roquefort cheese. Roquefort cheese. What industrially they are growing? Not industrially, of course, at our home also as a household product only we are using these Swiss cheese and Roquefort cheese. In the preparations of these two varieties of cheese also we are using some microorganisms. Have a look into all these different okay, food items what we are preparing by using microorganisms. In that, first one is convert milk to curd. How we use these microorganisms to convert the milk to curd? Okay, some environmental conditions are needed to convert the milk into curd by microorganisms. Here, what is the microorganism play pivotal role to convert the milk to curd is, okay, lactobacillus. What is, students? Lactobacillus or lab means what it is? Lactic acid bacteria. Lactic acid bacteria. Okay. Lab means what, Nana? Lactic acid bacteria. Okay. This is the bacteria which is going to convert the milk, okay, in the presence of temperature. What the normal temperature for growing bacteria? Well, you know very well. Okay, suppose to take for example 20 to 35 or 25 to 35 okay, degrees Celsius. It is the okay, optimum temperature where bacteria grow well. Okay. So this temperature maintained in the fresh milk, boiled milk, slowly the temperature, when you are going to boil the milk and keep it at one place, after that slowly after half an hour or 1 hour, slowly it is going to cool down. But when it is cooled down, when just suppose to think if it has maintained temperature 35 degrees, okay, 20 to 35 degrees or lukewarm milk, understood? That is the suitable temperature for adding or inoculate that small curd which contains lactic acid bacteria. Understood? Where they are going to perform the process of anaerobic respiration to increase their population, simply they are going to digest partially that protein material present in the fresh milk. The protein material present in the fresh milk, it is called casein. What is the protein? It is casein. protein casein. 82% of the, okay, 82% of the milk just we are observing casein is as maximum protein material which is going to partially digested by the lactic acid bacteria. Understood? So who are going to coagulate the protein material present in the fresh milk. So fresh milk plus lactic acid bacteria which is going to turn it into curd. Okay, when you are going to give sufficient time to the after the addition of lactic acid bacteria which is present in the small quantities of curd. So it is present in small quantities is enough. Small quantities of curd is going to be inoculate to this fresh milk. Okay, after a few hours, after 4 to 5 hours or 6 hours, slowly the lactic acid bacteria number increases in the fresh milk. In the fresh milk, the protein casein is partially digested. What happened, ma? Partially digested or partially coagulated. Coagulation, semi-solid material will be formed. Coagulated. That coagulated protein material slowly, okay, change the liquid state to semi-solid state. That is we are calling what? Curd. Complete curd formation we are observing. The curd, you know, what is the importance of curd? Every day we are using curd, but we must know the benefits of curd. And we must know the useful things what we are obtaining from curd. It is very important, you know. So generally the curd every day we need to use curd, okay, in the right and appropriate amounts because the curd contains. Now here we are discussing, Nana. So lactic acid bacteria is very important bacteria which is going to convert the fresh lukewarm milk into curd. Okay, the curd already contains lactic acid bacteria. When you are going to add little amount of curd to the fresh lukewarm milk after few hours, then that lactic acid bacteria can easy to digest partially that protein materials which they are naturally present in the fresh lukewarm milk. Then that partially digestion what it done in the fresh lukewarm milk then automatically turn into curd. Understood? One more time we better to write here. Okay, first suppose to take it is a fresh lukewarm milk. Lukewarm milk means little bit temperature. It may be maintained temperature what already we discussed like 20 degrees to 35 degrees Celsius is a suitable temperature in which bacteria can easy to multiply their number by the process of anaerobic respiration what it done here. Understood? So fresh lukewarm milk okay turn into curd. Now we better to discuss something about the useful things what they are present in the curd. Okay, curd is very much important. Every day we better to take sufficient amount of curd, appropriate amount of curd in our foods. So curd contains especially more proteins, proteins are made up of amino acids. Understood? Okay, in that total amino acids, some are there essential, some are there non-essential. Essential amino acids are nothing but the essential amino acids which we are only fulfill by taking diet only, through food. So here, suppose to take a person who are going to take curd regularly in the sufficient amount, then definitely they are maintaining sufficient amount of or quantities of proteins. Understood? So these are the more proteins we generally observing in the curd, and in the curd we are also observing very important vitamins. So many vitamins, but here we must discuss about vitamin B12. Vitamin B12. What is the chemical name of vitamin B12? You know, cyanocobalamin. Cyanocobalamin. Understood? It is very important for it is very important for stop that particular vitamin deficiency diseases like pernicious anemia. What it is, students? Pernicious anemia. Pernicious anemia means what it is, where the okay, blood contains blood is in the condition, or blood contains low amount of RBC, low RBC percentage. So in that this particular condition and the state of human being, it is called pernicious anemia. It is okay, somewhat danger. So it is going to be decreases or stopped by the sufficient amount of B12 vitamin which naturally available in the curd. Understood? And it is very important factor. Curd is a very important factor which is going to check every time pathogens. Whatever the pathogenic bacteria or pathogens which they are enter into our body, keenly, closely monitoring done by this, okay, all these are the materials which they are present here. Understood? Which contains lactic acid bacteria. Understood? So pathogens, okay, monitoring is also done by this. You know, right? So this is the very important features, generally aspects we are obtaining from the curd, what actually prepared by the adding LAB, lactic acid bacteria. Understood? Next one, this lactic acid bacteria, along with lactic acid bacteria and some other okay, members of yeast, you know, right? Saccharomyces cerevisiae. Saccharomyces cerevisiae is the yeast. Understood? It is a member of fungi. Okay, it is a type of fungi. Understood? So this is the yeast, also known as a baker's yeast and brewer's yeast. Baker's yeast means, you know, bakeries. In the bakeries, what generally we are observing, what they prepare? They prepare bread, buns, cakes, okay, pastries, like different food materials they are preparing. In the industrial scale, they are preparing, right, everything. In all the cases also we are observing the importance of microorganisms. Understood? And you know, right, in the beverage industries, that yeast, Saccharomyces cerevisiae is very important in beverage industries. So like that, okay, so many microorganisms, they are playing pivotal role in the, okay, household products, what generally we are obtaining. Understood? In the preparations of, along with these, we are also observing in the preparations of idli and dosa. Idli and dosa, they fermented, na? They fermented by bacteria. Fermented by bacteria for getting texture, taste, okay, and some porous properties we are observing. Okay, that porous properties generally we seen in these particular cases because of releasing carbon dioxide. Whatever the bacteria we are adding here, whatever the bacteria they fermented means what? Fermentation, is it aerobic respiration or anaerobic respiration? It is anaerobic respiration. But whatever it may be, they are releasing carbon dioxide. Whatever the gas is produced in the fermentation of all these food products, they are releasing, and they are trying to come out of the duff or come out of the raw material. What is the material we prepare to make idli, dosa, like whatever the foods we are preparing. So the duff contains much amount of bacteria which is going to ferment the sugars, and they are going to release carbon dioxide. Then these carbon dioxide is trying to come out of the duff, then it is giving some during the releasing gas carbon dioxide from the duff, it is going to produce some pores or some small holes. These holes are giving very much okay, what we call texture, very soft, okay, idli, dosa, whatever the in the bakery industry, what products they are obtaining, in all the cases, why should they are using? For the main important thing, it is for texture and taste. Understood? And some in cases flavor. So this is the important information what we are obtaining from the lab. And next one is, you know, okay, that is yeast. So whenever you are going to add this okay, yeast plus the bacteria which already fermented, means what? bacteria what already fermented bacteria present in a fermented duff is used to prepare bread. Okay. The bread preparation also we are in the bread preparations also we are using fermented duff already contain bacteria and yeast. Here the yeast is Saccharomyces. All of you note down Saccharomyces cerevisiae. Saccharomyces cerevisiae. Okay, it is a yeast, a fungal member. Understood? This is also very important for the preparation of bread, where the soft bread we are obtaining by releasing carbon dioxide gas during the process of anaerobic respiration done by these living organisms as microbes, which they are very much important for the production of all these household products. Okay, next coming to the next topic, next important food product, what it contains, these microorganisms like bacteria and yeast. We are going to discuss about some other products, household products, how do they produced in the presence of microbes. So in that now we focus our concentration on fermented fish. This bacteria we are also using for fermentation of fishes for increased shelf lifetime. Whenever we want to transport the fishes from one area to another area, okay, it may be taking time 2 days or 3 days. So without spoiling of all these fishes, they sustain their freshness just by using some bacteria which are going to fermented. Understood? To keep freshness long time until they are reaching to the consumer. So fermented fishes also done by the microorganisms. And the next one is fermented soybean. We are observing fermented soybean. Okay. Next one is fermented bamboo shoots. In so many areas, the people are using bamboo shoots as food. So just they are going to transport these bamboo shoots for long time from one region to another region. So they have to sustain their freshness in all the bamboo shoots. Before itself they have to do fermentation process. Then it can easy to maintain their shelf life. To maintain freshness in all the bamboo shoots, what the people are using bamboo shoots as food. So this fermentation process are needed in all these different kinds of food products. What actually we are going to do the fermentation process by using microorganisms. Now got the point, students? So next one, we are also using bacteria. We are also using these particular microbes in a toddy drink. The preparations of toddy drink. Understood? What is toddy drink? Already we little bit discussed. This is called palm sap. You know very well, very long palm trees. From that only some people they are going to tie some pots to collect the liquid what it comes from the plants, palm trees. Simply they are collecting it and ferment it. So to fermentation of this particular liquid, what we are calling palm sap, to maintain taste, just we have to ferment the palm sap. That is the drink what it is called toddy. This is very much important a variety of drink for South Indian people. South Indians. In the South Indians, majority of the cases they are using this drink. That is a toddy drink. To ferment the toddy drink, we are using microorganisms. Understood? And the next one, it is we are also using these particular microbes in production of varieties of cheese. You know what is cheese? I think so many people, they have very much interest to take food cheese. Yes, right? So this is a cheese, okay, based on their characteristic features which we see for taking cheese as much as with very much interest. Okay, especially for texture, we want to take good texture, and also we want to take okay, good flavor. Okay, we want to take what it is, a taste, good taste. So all these are the main important okay, characteristic features which distinguish the different kinds of cheese. Understood? So in that two varieties of cheese we are generally observing, that is okay, Swiss cheese. You know, already I said cheese is an ancient or oldest food variety, okay, of all these people, right? You know, right? This Swiss cheese is a variety, one of the varieties, okay, which maintain all these characteristic features like texture, flavor, taste. To obtain all these different kinds of characteristic features in the Swiss cheese, we better to add some microbes. Understood? Especially the Swiss cheese produced by means of all these important favorable factors like texture, flavor, taste. Okay, if you want to get, we better to add bacteria, that is called Propionibacterium shermani. Propionibacterium shermani. Propionibacterium shermani is a bacterium which is very much important and which is very much used to preparation of Swiss cheese. Right, children? So Swiss cheese preparation is very important by adding a bacterium called Propionibacterium shermani. This Propionibacterium shermani is going to add all these different kinds of interests to take cheese very much. Understood? Like texture, flavor, taste, all those are maintained by, okay, releasing some different kinds of chemicals into the cheese preparation. Then it is giving all these different interests. Understood what actually we are obtaining from the Swiss cheese? In the Swiss cheese, whenever you are going to observe in the Swiss cheese, okay, more or large holes we seen. These large holes we seen because of releasing a gas called, it is very important bit, called CO2. So during the process of respiration, what it is conducting by that particular okay, bacteria, what we are adding here. Understood? So it is simply releases CO2. CO2 gas is going to make very big and large holes in the cheese. So then that cheese giving some properties or features like softness, texture, flavor, taste, everything. And one more variety of cheese we are observing, so that is that is Roquefort cheese. The next variety of cheese, that is Roquefort cheese. This is also a type of a variety of cheese. So Roquefort cheese is produced by, Roquefort cheese is also fermented and giving okay, especially for flavor. We are going to grow this particular cheese. We are growing a variety of microorganism called a fungus. We are growing fungus, a variety of fungus on Roquefort cheese. After the preparation of Roquefort cheese, if you are going to use some fungal members, if you want to grow some varieties of fungal members on this Roquefort cheese, okay, gives a very good flavor. Okay. That fungus is, what is the fungus it is? The fungus is a Penicillium roqueforti. Penicillium roqueforti. It is the okay, fungal organism what we are growing on the Roquefort cheese, which gives a very good flavor to take food sufficiently, to take very much interest. Okay.